Welcome back to G Fun Facts Online. Today we are diving straight into one of the most profound public health crises in human history, the silent pandemic of antimicrobial resistance, commonly known as AMR. In the year 2019 alone, antimicrobial resistance was directly responsible for approximately 1.3 million deaths globally. To put that staggering number into perspective, that is a death toll greater than HIV/AIDS or malaria. And if current global trends hold, that number is projected to skyrocket to a devastating 10 million deaths annually by the year 2050. The core of this problem lies in a relentless evolutionary arms race. Pathogens are mutating and sharing resistance genes with each other much faster than our traditional pharmaceutical pipelines can develop new drugs. But crucially, they are mutating faster than conventional clinical laboratories can even diagnose them. For nearly a century, clinical bacteriology has relied on the exact same fundamental principle. You take a sample, grow the bacteria in a petri dish or a liquid culture, expose them to various antibiotics, and simply wait to see what survives. While this gold standard method, known as antimicrobial susceptibility testing, is highly accurate, it is agonizingly slow. It often requires several days just to yield actionable results. Now, imagine being a doctor in an intensive care unit. An infection can cascade into lethal sepsis within a matter of hours. Doctors do not have days to wait for a petri dish to grow. Because the clock is ticking and a patient's vitals are dropping, physicians are forced to make educated guesses. They often prescribe broad-spectrum shotgun antibiotics, hoping one of them hits the target. This is where the massive biological bottleneck occurs, and it is a bottleneck we desperately need to fix. Let us examine the limitations of this status quo in more detail. The traditional workflow for diagnosing a bacterial infection involves several highly time-consuming steps. First, sample collection. Blood, urine, or tissue is drawn from the patient. Next comes isolation and culturing. The sample is incubated in a warm environment until the bacteria replicate to a detectable threshold. Because bacteria multiply through cellular division, this takes time. For slow-growing pathogens like Mycobacterium tuberculosis, the bacteria responsible for TB, this step alone can take weeks. Finally, there is phenotypic testing. The isolated bacteria are exposed to a panel of antibiotics at varying concentrations to determine the minimum inhibitory concentration. This is the absolute lowest concentration of a drug that prevents visible bacterial growth. This traditional method evaluates the phenotype, which is the observable behavior of the bacteria. The massive problem here is the sheer biological time penalty. While patients wait, they receive empiric therapy. If the bacteria are resistant to the empiric choice, the patient deteriorates rapidly. If the bacteria are susceptible but could have been treated with a milder, highly targeted drug, the highly potent, broad-spectrum antibiotic unnecessarily slaughters the patient's beneficial microbiome. Even worse, it exerts selective evolutionary pressure on all surviving bacteria in the body. By wiping out the weak bacteria, we leave behind only the strongest, most resistant ones, effectively breeding new superbugs right inside the hospital. Enter algorithmic bacteriology. This is a radically new paradigm that merges microbiology, high-throughput sequencing, digital microscopy, and advanced machine learning architectures. By teaching artificial intelligence to read the genetic fingerprints, protein signatures, or physical behaviors of bacteria, scientists are condensing the diagnostic timeline from days to mere minutes. Algorithmic bacteriology completely bypasses the wait time by analyzing the pathogen's intrinsic data and running it through predictive algorithms. This incredibly exciting interdisciplinary field relies on three primary data streams. First, genomics, analyzing the DNA sequence of the bacteria to find hidden resistance genes or mutations. Second, proteomics and metabolomics, using highly sensitive mass spectrometry to analyze the molecular composition and protein makeup of the bacteria. And third, microscopy and imaging, using high-resolution optics to observe how individual bacterial cells react to antibiotics in real time. Machine learning sits at the exact center of this web, because biological data is impossibly vast and complex. A single bacterial genome contains millions of base pairs, and a single mass spectrometry reading contains thousands of individual peaks. Traditional rule-based software cannot easily parse this sheer volume of data. Machine learning models, however, excel at finding hidden patterns in high-dimensional data. Let us look closer at genomic-based machine learning models, which involve reading the biological blueprint of resistance. The most prominent area of algorithmic bacteriology involves whole genome sequencing. As the cost of sequencing has plummeted over the last decade, it is now possible to sequence a pathogen's entire genome within a few hours. This genomic data is translated into mathematical features. These might be k-mers, which are short, fixed-length sequences of DNA, or unitigs, which are assembled sequence fragments, or single nucleotide variants, which are essentially single-letter typos in the DNA code. These features are fed into supervised machine learning models that have been trained on vast, global databases of known bacterial genomes, paired with their laboratory-confirmed resistance profiles. Historically, genomic AMR prediction used simple, rule-based or lookup table approaches. If a specific resistance gene, like the MECA gene in MRSA, was present, the bacteria was automatically flagged as resistant. However, biological resistance is rarely that simple. Resistance can emerge from the complex interplay of multiple subtle mutations, changes in how genes are expressed, or novel evolutionary adaptations that are not in any existing database. Machine learning algorithms, such as random forests, support vector machines, gradient boosting machines, and deep neural networks, do not rely solely on predefined human rules. They independently learn the complex mathematical associations between genetic sequences and clinical resistance. A prime example of this evolution is the Group Association Model, or GAM, developed by researchers at Tulane University and published in early 2025. The researchers tackled two notoriously difficult pathogens, Mycobacterium tuberculosis and Staphylococcus aureus. Traditional genomic tools frequently miss rare mutations or falsely flag completely unrelated genetic quirks as resistance markers. The GAM approach, combined with machine learning, completely bypasses the need for prior expert knowledge of resistance mechanisms. By comparing the whole genome sequences of massive bacterial populations with varying resistance patterns, the AI learns to identify the exact genetic changes that reliably indicate drug resistance. In clinical validation studies utilizing samples from China, this machine learning-enhanced model drastically outperformed standard World Health Organization prediction methods, especially in forecasting resistance to critical frontline antibiotics. But bacteria evolve incredibly rapidly, a reality that frequently causes rigid diagnostic models to fail when they are introduced to new geographic regions or novel strains. In January 2026, researchers from the Okinawa Institute of Science and Technology published robust machine learning models designed specifically to capture the rapid evolution of antimicrobial resistance in Staphylococcus aureus. Trained on a massive global database of genomes, the algorithms proved capable of accurately predicting specific resistance profiles even when analyzing messy, incomplete sequence data. More importantly, the models maintained their reliability when confronted with genetically distinct strains they had never encountered during training. This demonstrates a level of generalizability that is absolutely critical for real-world clinical implementation. While genomic sequencing is incredibly powerful, it tells us what a bacterium is capable of doing, not necessarily what it is actually doing in a living host. Sometimes a bacterium possesses a resistance gene, but the gene is turned off, meaning it remains unexpressed. To capture the true phenotype without waiting days for a culture to grow, algorithmic bacteriology has turned to computer vision and deep learning. This brings us to what scientists call the 30-minute miracle. In a landmark November 2023 study published in the journal Communications Biology, researchers from the Oxford Martin program on antimicrobial resistance testing successfully combined fluorescence microscopy with artificial intelligence. Their approach operates at the absolute smallest scale, the single-cell level. When bacterial cells are exposed to antibiotics, they undergo microscopic structural changes. For example, there are rapid alterations to the bacterial chromosome long before the cell actually dies or stops dividing. The human eye cannot easily quantify these subtle, rapid shifts under a microscope. However, deep learning models, particularly convolutional neural networks trained on thousands of bacterial cell images, certainly can. The Oxford team tested clinical isolates of E. coli against antibiotics, like ciprofloxacin. By analyzing the structural changes inside the cells, the AI could determine whether the antibiotic was successfully disrupting the cell, which indicates susceptibility, or if the cell was completely ignoring the drug, which indicates resistance. The results of this study were absolutely staggering. The AI achieved at least 80% accuracy on a per-cell basis, delivering definitive AMR detection in as little as 30 minutes. This is an order of magnitude faster than current gold standard methods. It transforms a multi-day wait into an immediate, actionable result that can directly guide emergency room prescribing in real time, potentially saving countless lives. Another massive leap in algorithmic bacteriology is occurring right now in the realm of mass spectrometry. Matrix-assisted laser desorption ionization time-of-flight mass spectrometry, commonly known as MALDI-TOF, is already a staple in clinical microbiology labs around the world for identifying bacterial species. It works by firing a laser at a bacterial sample, creating a spectrum of peaks that acts as a unique protein fingerprint for that species. However, using this technology to detect actual antibiotic resistance has historically been incredibly difficult, requiring meticulous, manual pre-processing of the data. Recent advances have solved this by applying deep neural networks directly to the raw, unedited spectra. The MS-Deep AMR architecture is a prime example of this breakthrough. Utilizing the public DRI-AMS database, which contains over 300,000 mass spectra and 750,000 antibiotic resistance profiles, researchers trained MS-Deep AMR to predict resistance in deadly pathogens like Escherichia coli, Klebsiella pneumoniae, and Staphylococcus aureus. The deep learning model achieved an area under the receiver operating characteristic curve of over 0.83, which improves upon older, traditional machine learning methods by over 10%. Crucially, the researchers employed a technique called transfer learning. Typically, machine learning models suffer a massive drop in accuracy when moved from the hospital where they were trained to a completely new hospital with different equipment or different local bacterial strains. By allowing the MS-Deep AMR model to adapt slightly to a very small amount of external data from a new laboratory, its predictive accuracy skyrocketed by up to 20% compared to models trained purely on external data. This proves that powerful AI models can be pre-trained on massive global datasets, and then easily fine-tuned for local low-resource laboratories that simply cannot afford massive data collection operations. Algorithmic bacteriology is not just about laboratory diagnostics, though. It extends directly to the patient's bedside through clinical decision support systems. The ultimate goal here is precision stewardship, which means giving the exact right drug to the exact right patient at the exact right time. Researchers at Stanford Health Policy, including Dr. Jonathan Chen and Dr. Mary K. Goldstein, have pioneered the use of AI to generate predictive antibiograms based on electronic health records. A traditional antibiogram is basically an annual hospital report showing overall bacterial resistance rates, but it is generalized and not specific to an individual patient. The Stanford team utilized machine learning to analyze the electronic health record data of thousands of patients. They factored in complex variables like medical history, past prescriptions, prior infections, and even geographical data. By synthesizing this massive web of information, the AI predicts the specific antibiotic susceptibility of the infection currently residing in a specific patient's body. This predictive capability allows doctors to put down the broad-spectrum giant shotgun and instead pick up the targeted small scalpel. Furthermore, by geocoding patient data in regions like the Dallas-Fort Worth area, the Stanford team mapped the social disparities of antimicrobial resistance. They discovered that drug-resistant organisms, including life-threatening MRSA, densely cluster in areas with high socioeconomic deprivation. This powerful finding proves that AI can act not only as a clinical tool for doctors, but also as a highly advanced epidemiological radar, directing public health resources to vulnerable neighborhoods, long before superbugs can spread further into the community. Beyond just diagnostics and prescribing, there is another major problem. Because of low profit margins and high failure rates, major pharmaceutical companies have largely abandoned antibiotic research and development entirely. The pipeline of novel antibiotics is running dangerously dry, while antimicrobial resistance continues to rise year after year. Here, machine learning is stepping in as an incredibly powerful engine for drug discovery. Traditionally, finding a completely new antibiotic required scientists to screen thousands of soil samples or synthesize chemicals in a painstakingly slow trial-and-error process that took decades. AI models, particularly graph neural networks, can now computationally screen databases of millions of chemical compounds in a mere matter of days. In groundbreaking studies, researchers have applied deep learning to massive chemical libraries, such as the Zinc-15 database, which contains over 107 million unique molecules. By teaching the AI what biological features make a molecule toxic to bacteria but safe for human cells, algorithms have successfully flagged entirely new, structurally distinct classes of antibiotics. These AI-discovered drugs operate via molecular mechanisms never before seen by the medical community, allowing them to bypass existing bacterial resistance entirely. Furthermore, machine learning is being used to design and optimize antimicrobial peptides. By analyzing the sequence of natural immune peptides, generative AI models can actually engineer synthetic, hyperlethal peptides that punch physical holes in the cell membranes of drug-resistant pathogens, offering a completely new therapeutic arsenal in the fight against superbugs. Despite all of these profound successes, the integration of algorithmic bacteriology into frontline healthcare still faces significant hurdles. The first major hurdle is the explainability problem. In the field of medicine, saying, "because the computer said so," is simply not an acceptable reason to risk a patient's life. Many deep learning models are what we call black boxes. They provide highly accurate predictions, but the internal logic used to reach those mathematical conclusions is completely opaque to human understanding. If a model treats genetic mutations as isolated mathematical variables without considering their actual biological function, it risks making catastrophic errors when novel mutations arise. To earn the trust of clinical microbiologists and doctors, researchers are aggressively developing explainable AI that can map its mathematical decisions directly back to known biological and biochemical pathways. The second hurdle is data bias and class imbalance. Machine learning models are ultimately only as good as the data they consume. Currently, the vast majority of genomic data in the world comes from wealthy, industrialized nations. If an AI is trained exclusively on bacterial strains from North America and Europe, it may fail catastrophically when deployed in sub-Saharan Africa or Southeast Asia, where completely different AMR mechanisms dominate the landscape. Furthermore, because highly resistant superbugs are statistically rarer than normal, susceptible bacteria, the datasets often suffer from class imbalance. If a model is fed 9,000 susceptible genomes and only 1,000 resistant genomes, it may artificially skew its predictions towards susceptibility, which could be deadly in a clinical setting. The third hurdle involves standardization and regulation. Clinical diagnostics are incredibly highly regulated for good reason. The Food and Drug Administration and the World Health Organization require rigorous standardized proof of efficacy before any AI tool can be legally allowed to dictate patient care. Creating theoretical frameworks for the continuous regulatory validation of AI algorithms, which by definition change and update as they learn, is a monumental legal and ethical challenge that policymakers are currently scrambling to solve. As we move deeper into the late 2020s, the future of algorithmic bacteriology relies on hyperconnectivity and global collaboration. To overcome the major issues of data bias and patient privacy, researchers are fully embracing a concept called federated learning. Instead of sending highly sensitive hospital data to a central, global server, the machine learning model itself is sent directly to the local hospitals. The model trains locally on the hospital's private data, learns the new resistance patterns, and then only sends the updated mathematical weights back to the central server. This incredible technique allows global healthcare systems to collaboratively build an unbelievably powerful, diverse AI without ever sharing an individual patient's protected health information. Furthermore, the future will see the integration of AI with Internet of Things diagnostic devices and point-of-care biosensors. This will completely democratize the technology. Imagine a small handheld device in a rural, low-resource clinic that can analyze a single drop of blood, sequence the bacterial DNA, run a lightweight neural network locally on the device, and prescribe the exact antibiotic needed. all within a single hour. For decades, humanity has been slowly losing ground in the invisible war against antimicrobial resistance. The bacteria have constantly had the ultimate advantage of rapid evolution, constantly outpacing our slow, analog diagnostic techniques. Algorithmic bacteriology finally levels the playing field. By harnessing the massive computational power of machine learning, deep neural networks, and artificial intelligence, we are completely transitioning from a slow, reactive, trial-and-error approach to a proactive, highly predictive science. AI is not replacing the microbiologist or the physician. Rather, it is providing them with a profound biological superpower. From the 30-minute microscopic detection of resistance to uncovering entirely new classes of life-saving drugs, machine learning models have become the absolute vanguard of infectious disease management. If the promise of precision stewardship is fully realized, we can preserve the miraculous efficacy of our current antibiotics, dramatically slow the birth of new superbugs, and entirely prevent the looming specter of a dark post-antibiotic era. The algorithms are learning every single day, and with them, our capacity to protect human life is evolving. Thank you so much for joining us today on G Fun Facts Online. We hope you enjoyed this deep dive into the fascinating world of algorithmic bacteriology. Keep learning, stay curious, and we will catch you in the next episode.